Hello, everybody.
Hi, Matt. I think
Hello.
I think almost everyone's here. I think we're just waiting for Kimberly as well. Then that should be everyone.
Advance apologies. I've got to click off the call at 12:30. I have to go to something else.
Yeah, and we have a meeting in an hour time as well. So maybe we simply just start.
Yeah, I reckon.
Yeah, that's fine. Oh. All right, Kimberly's here, so yeah, we're all good to start. So I think it's just a quick one today, brief update from everyone for the past month, I guess. Hi, Kimberly. You're good with minutes? All right.
So who wants to start?
All right. I can start. I'm happy to start. So. All right, so yes, I've been doing synthesis. These are kind of like the compounds that I've already showed, uploaded onto GitHub the other day. I'm currently working through probably half of them already. So kind of this week I'm on the halfway mark to getting these compounds. So I've already added. So I'm still waiting for some of these. Green yet to arrive. So I bought the green yet, so the one in red to put to react with the ketone here. So these two green yet are still kind of like in the mail, and I've already ordered this one as well. So I think this was what Anthony was suggesting that I should also be doing as well. So I've got that order. So currently working through the middle here, and this week I think I'll try to get this lot done. And also, if the other one hasn't arrived yet, I'll do a just do some write up to catch up because I'm a bit behind with writing up. So I'll do some of the write up and I'll put that into Wilson's document to consolidate everything together. So yeah, that's what I'm up to.
So are all the ones on the screen targets though, right? None of them have been made yet?
No, they haven't been made yet. So these
okay,
are
they have your codes on there, obviously. So I assume the ones with the codes may be the ones you got already.
So these are the one I haven't got the code yet. I haven't actually prepared hierarchies for them yet. So that's why they don't have a name yet. But
Okay.
yeah, the rest of them should. So the one that has this code here is from the OSN original list. But these other ones are just what I'm making that are not from the list.
Okay, and just I mean, because I guess just coming back to the codes idea that we were talking about last time, I guess once they're made, then we need to assign them some kind of, unique MycetoS code, like what we were talking about last time. I guess that's the point at which it needs to go into the sheet and have the one MycetoS code. I would guess, because then as soon as we talk about them in future meetings, we have that unique identifier.
Yeah, a harmonized code, if you will. So yeah, Kimberly, do you have any update on your end from our? Okay, good.
Yeah, sorry, I've been way too busy with a lot of other things this month.
All right, no problem. So yeah, that's all from you, Sid, I guess. Matt and Dimitri, do you want to start next?
Yeah, sure.
Oh, let me start share. I forgot. Sorry.
Can you see my screen?
Yep.
Yeah. So basically, a short update on my work. I did like 3 more analogs from the OSN list, and we received 1 phenomenal from the Seven Oaks school. And all of them like are in different phases of characterization and purification, basically. And we're working on the Aldexime series as well, and there are 2 different protocols that I'm trying to implement. And probably I'll have some results by the next meeting, that's for sure.
So I guess as soon as we, again, the same thing applies here. So the internal codes are no problem because if things are being made, but as soon as they get made and all the characterization is done, so we're confident that the product is real and ready to go, then we need to assign it a MycetOS code. Yeah.
Actually, Matt, we had this conversation with you back in the summer, and I had some proposals like back then, but we never actually discussed this. So I think the best thing to do is probably just I'll show you what are the options that I came up with, and maybe we can report this to in the next meeting just to finalize like what will be the final convention system for the coding.
Yeah, there's one on the GitHub already, right? I mean, we went through the sort of consultation about what will be useful, and we're using this very similar protocol for the open source antibiotics, which accounts for batch numbers and salts and all that kind of stuff, and it's pretty comprehensive. And I think it's a good one. We would just need to
MycetoS coding system.
Yeah.
Yeah. I think that's the best option as well.
Yeah. So that what that needs though is for us to go into the spreadsheet of mycetoma molecules and just assign a bunch of codes. And we may as well start at the top of the sheet and work down. So that when new molecules it gets into the sheet, and it means there's no duplication of the numbers.
Yeah. Because there has been some duplication in the phenomenal analog Google sheet as well, I've noticed. So this has to
okay, but we can deal with that at the same time.
Yeah.
What's the status of the 7Oaks molecule? What does it look like?
I'm not entirely sure because I couldn't confirm its identity on LCMS. I haven't looked at the NMR yet. I mean, I actually have the proton NMR, but it doesn't look very good, so I'm not sure. Probably I'll have to ask the person from Seven Oaks just to confirm that there hasn't been like any error with the coding system.
But it needs purification, does it?
Yeah, absolutely. It has like
at
least like four peaks on LCMS, so it definitely needs purification.
Okay, right. And then after that we can characterize it.
Yeah, but I'm already working on this basically. All of them. So all of those 4 compounds that are in the list have been made. Just catching up like on different stuff like characterization, like extra NMR and other information like that.
And again, just to re-emphasize the point last time that I think that all of these things, it would be very helpful if when we have these structures, we just keep an eye on the predicted log D. You know, again, when we come to write up a paper on this stuff, we're going to have to make the case that we are taking into account what we learned last time. And what we learned last time was that certain log D values are more promising for a potential in vivo treatment than others. So if we end up with a bunch of molecules that we're making that all have, you know, the log D value that we don't want, we're going to look a bit strange.
So does that imply that we should review the whole OSN list and take out compounds like in the future that have like very high log D values?
No, we need the range.
Should we define like what range do we want and maybe then take out like that? Because the ones, the DM31 and 31.1 have been on the OSN list before. So I didn't come with this idea by myself.
No, the list is good. I just think for the molecules that we're making, we need to make sure we're covering the range, right? So that we take into account the hypothesis that we had last time.
Okay.
Yes. Do we actually? I don't think we've discussed about this. But do we have like we haven't agreed on like a one software or system to do the log D? Because for me, when I was doing mine, I just used Chemaxon because it was free. But I think when Ben was doing that log D, he was using a proprietary version of.
It was using
how to calculate log.
was
Yeah.
using Stardrop, yeah. And we agreed that he would be, because he updates the list all the time, so he would be putting his Stardrop values in the actual scheme.
Okay.
Because
Okay.
otherwise there's a deviation in the numbers.
Yeah. Okay. Yeah, that's good.
Depending on the software that you're using.
Okay. Yeah. If he's doing it, then it's fine. I don't have to worry about it too much.
Well, I mean, like if he wants someone else to do it, then we need just to decide like what software we're using, and everyone just keeps with the same software.
I have another question. With the characterization of these molecules, do you also take into account how stable they are when they are dissolved in DMSO? Because that's how we test them. And we found some issues when we repeated something after a while. Not always they were reproducible.
Yes, Wendy. So I came across this problem just last week when I was catching up on the data points for one of the analogs that I've made before. And basically, I think that one of the compounds has degraded, unfortunately. At least I couldn't get like a normal carbon-13 NMR for that. Same applies for protein. So I'm not sure what happened there. And
which compound is that, Dimitri?
That's DM11.2. It's been made like ages ago. It was one of the first batches that has been tested, like that we've sent from UCL.
Yeah.
Yeah, because the problem is that for us there's a lag phase between in vitro testing and in vivo testing, and we cannot weigh a very small number, so we simply dissolve it in DMSO. So for us it's good to know how stable a compound is, because if they were tested in the beginning for the in vitro susceptibility, and we are currently doing in vivo testing, and if the molecule was not stable in DMSO, that might mean that we will get negative results in vivo, not because the molecule doesn't work, but because it was unstable and was degraded. So that's something we should take into consideration.
How do you store them? How do you store the molecule? So it's in DMSO, but what's the temperature, concentration, air?
Wilson, where did you store them?
Well, after sort of reconstituting them in DMSO, we put them in -20. And when we use it, it's 4 degrees. We store it at 4 degrees, and then, yeah.
Okay, and what's the
concentration is 10, we do it to 10 micromolars.
10 micromolar, and do you expose it to air, or is it sealed?
Sealed.
Okay, so we could easily test that by just taking as one of the samples that we're worried about, putting it in deuterated DMSO, keeping it under the same conditions, and then periodically taking an NMR?
Yeah.
So that's a simple one.
Most of them should be stable. Most of them should be stable, I think. But we need to test that, just to make sure.
Most of them should be. Chris Rice is on the call, who did the amoeba screen. Chris, didn't you have a problem with freeze thawing, or was that unrelated?
That's probably unrelated, but we see this with the azole class in general, that they never survive freeze thawing. But that was in a media, so it wasn't with our stock DMSO, but we have seen this sort of before, where if you are storing azoles in a base media, then they tend to precipitate and be less soluble. But for your in vivo things, what we've tried to do is, if we do an in vitro assay and it's out with 3 months of reconfirmation, we generally either ask for a resupply from the chemist so that that stock is then fresh. We then do a secondary run of confirmation in vitro to determine the IC50 again, and then from that we then determine what the best dosing regimen is for our in vivo studies. So that's how we try and get around potential insoluble molecules that have been stored for long term.
So yeah, so for me, I have actually encountered any my compounds, especially the one that from 3 years from 2017 when I was doing my honors. So I stored them in under nitrogen sealed in the fridge for a couple of years, and they're still quite stable. As I've only done a characterization for some of them, but I haven't actually taken into account like the storage under in DMSO condition yet. But is it also possible if to kind of like work around the problem? Is one solution is I can also send extra sample as well, like more than needed, if that's possible, or do you think that's yeah, I don't know.
I think that's a good idea.
So instead of sending 3 milligrams, I can send 6 milligrams, for example, where possible.
I mean, it's not the amount that we are worried about. It's that we don't know that, or there's some compounds that we can't reproduce results like what we've got the first time.
Would you be able to specify on GitHub with which ones you had a problem?
Yeah, I'll have a look into it, but yeah, I'll have a look into it. And also with one of Hong's compounds, I realized that it sticks to plastic. It's 9.1. It gives us a slightly yellow tinge, and it sticks on the Eppendorf tube. Okay. I'm not sure if I haven't. I realize this when I'm doing the in vivo work, and I'm not sure about the rest. From the panel of the in vivo work, this was the only one that stick to the tube. I'm not sure about the rest because we're not trying the in vivo, but yeah, something to think about.
I think the stuff you've done, Hong, about re-examining compounds at a couple of years old is very important, and that's useful. I think we just need to replicate the conditions that Wendy and Wilson are using. And if we, because we happen to use deuterated DMSO as a solvent for NMR, it's a really simple experiment to do, just keep it in the tube, and periodically NMR it again and see if it survives. I guess, I mean, they're looking at the structure. There's obvious ways in which this thing might be acid sensitive, and you imagine it in a reasonably high concentration, there may be a way for it to fall apart a little bit. But given that we're basing this on phenarimol, which is a commercially available product, I wouldn't have thought that stability would be a huge issue. That's not to say that all the analogs will be equally stable, obviously.
Yeah. So do you think, Matt, that how many of, because I'm obviously making quite a bit there, so I could take a random few, or I can keep track of all of them as well. I don't know, like, what do you think is like the optimal amount in terms of like effort and value for my time, essentially?
Yeah, no, if there's a molecule that Wilson and Wendy think that there may have been a problem about with reproducibility, let's take that one and then just put 10 mgs of it in deuterated DMSO or something at a concentration that roughly matches theirs, and keep it in a way that roughly matches theirs, which of course would be a NMR tube with a tip on it, with a top on it, and take an NMR like after a month or something, and see what happens, and see if we see any evidence of that. It's convenient that the solvents will match if we use deuterated DMSO. So I would just say one sample that we think maybe have an issue, that maybe we think has an issue, and see if we see anything.
If you could just tell us the name of the compound that he has problems with, so we can test it.
Yeah, I think we will post that on GitHub. One of the compounds was the EPL compound 495, wasn't it?
Yeah, 14.1 Hong's compound. That's based on 0495 EPLBS.
All right, I'll keep track of that one.
Okay.
So Wilson, one thing to also consider is the types of tubes that you're putting these things into. If it's polystyrene or polypropylene, then both of them have different binding affinities when you're trying to solubilize your drugs. We've noticed with some of our compounds that we've had to move more to a polystyrene tube when reconstituting it for our in vivo studies. So then the other question with that comes is, what are you using for your in vivo studies, and where is it getting administered to? Are you doing IV, IP, oral? Are you using PEG? Are you using PBS? Like, there's so many variable factors into what could be used as a vehicle to then progress it into animals, and the solubility issues with each of all of those vehicles that I just mentioned as well. Okay, I'm not sure what sort of plastic we're using at the moment, because we're using a few in the course of the whole infection. Definitely something that we need to sit down and think about. Yeah.
Yeah, typically speaking, in the chemical synthesis, typically speaking, we don't have anything really come into contact with plastic. It's all glass and borosilicate and stuff. So that would be something we wouldn't necessarily pick up on in the chemistry lab, I would say. Yeah, but we know that as soon as it goes into biology, it comes into contact with all kinds of things. And you know, Eppendorf's, for example, we just don't use really in the lab, in the chemistry lab.
Yeah, most of my reactions are also in glass as well, so I don't actually use plastic in my chemical reaction at all. So
do you use glass syringes?
Sorry,
Hank, do you use glass syringes as well?
Yes, glass syringes as well.
Okay, something to be aware of. Useful.
But regardless of the problems, we do have in vivo results. So maybe Wilson would like to share those.
Yeah, I'm on tenterhooks. I've got to go in 7 minutes, and then.
All right, hold on. I am trying to get this online. Screen.
Is the project finished? Have we succeeded? Is it all over? We found the perfect molecule.
Can you see my screen? Yeah, okay, so we have about 14 compounds, if I counted it correctly, that we are doing in vivo work on, and these are the MycetOS for that, and we chose ones that are below 4 micromolars. So for DM7.1 and 0549 EPL ones, MycetOS has to be determined, yet we missed this compound because it got sort of lost in a big Excel sheet. We didn't really notice that the IC50 was at a good value. So, but we're currently doing this in vivo at the moment, and then if the in vivo work goes well, then DM7.1 and BS0549 will go back into doing the MIC50s. So this is the first sort of in vivo result that we have got. Made it into 3 figures because it's then easier for you to see. As you can see here, the compounds are sort of improved larvae survival. There's 1, 2, 3, 4 of them. 1 in figure A, 2 figure B, and 1 in figure C. Yeah, they are highlighted in bold, basically. So
what you see in this figure, it's the dashed line, that's the non-infected larvae. So that's a quality control. And the black line, that's the larvae infected but not treated with an antifungal agent. And as a control, we use itraconazole as well. And as Wilson said, so if you look at the black normal line, you see that most of the larvae have died at day 5. Only 1 or so is remaining until day 10. And if you then compare it with the green line in figure A, you see that that line is higher. So that means that larvae start to die at a later time point. so the AUC under the curve is larger than the one from the controls, and that indicates that this compound is able to prolong larval survival, because roughly 20% of the larvae is still alive at the last day. And if you look at all the figures, especially compound DM7-1 looks very promising, because you see that there we have a high survival and that the line is really shifted towards the longer survival.
Better than itraconazole.
Yeah, because itraconazole doesn't prolong larvae survival. In this experiment, it actually looked more promising than in the previous experiment, but in general, we never see much effect of itraconazole.
Do you have a positive control for this assay?
Yeah, amphotericin B, we are included that in the second round as a positive control. That always prolongs larval survival. Not to 100%, but it does prolong larval survival.
That's great. Are you excited?
This is only one experiment at the moment. We are currently running a second and a third one.
Would the next step be to compare the two, DM7-1 and AMP-B?
Sorry, DM7.1N?
And compare it to itraconazole and AMP-B, the two already on the market drugs?
You mean in vivo or?
Yeah, in larvae, yeah, in vivo.
Well, we are already doing that at the moment.
Yeah, you already did, yeah.
But yeah, this is okay. Anyway, running the second and third experiment, and then results we'll get sometime between the end of the week and start of next week, and then update the results and put it on GitHub.
That's
Hopefully,
great.
we get the same trend.
Mm-hmm. Sure.
Yeah.
So Wilson, this is some great results. Can I ask a couple of questions?
Yeah.
The first one being, what concentration were all the drugs performed at in this study? And then second of all, with DM7, rather than a question, more of a comment, that with this single concentration of DM7, should you then now move into doing different dose responses to tease out what is the maximum or minimum response of the compound to then give you the best larval survival?
Concentration, oh God, I can't remember by head. Wendy, how much per larvae? I just can't get it in my head at the moment. But we do 3 treatments, 4 hours, 28 and 52 hours, our infection. We injected, I think it was 20 micromolar per larvae. Yeah. That's the concentration of drugs we inject into the larvae. So over 3 days. Yeah, so was that your question? Yeah. Have I answered your question?
Yeah, so we do the maximum we have. And of course, we love to do, we would like to do dose response curve, but the larvae season will end within a few weeks' time. So I'm not sure if you will able to do that. But for now, this compound DM7-1 looks the most promising. But of course, this is only NS1, so we need to repeat to confirm. And that's what we're currently doing.
Yeah.
Sorry to butt in again, but has any work been done with the other pathogens besides the amoeba screen that Chris Rice gratuitously gave to us? Like the other mycetoma causative agents, have they been screened yet, or?
No, not yet. Yeah.
Okay.
Yeah, due to the long culture times, they need to be subcultured first and then processed. So that will take, one round will take a month. So yeah, we simply need to subculture them.
To see if these will hit other things.
Yeah.
Because you'd think with the MOA, CYP51.
One step at a time, though, I think, right? Let's confirm the results, and then we can think about other things.
So yeah, again, sorry, the we're having a bit of disruption in larvae supplies due to COVID. That's why I'm running a bit late. Also, the third and fourth experiment are running currently, and the manuscript has now been updated and to the current progress, and it's on the Google Docs. The link is on GitHub. And that's all from me today. That's the update that we have from Erasmus. Yep. Yeah, that's it for me.
All right, thanks, Lisa.
Do you guys get a chance to do any of the in vivo work with the pandemic box, or time's just running short?
We will be running the pandemic box this Friday, actually, on in vivo. Yeah, we wanted to finish the phenomal analogs first before moving on. Yeah.
Because normally around this time of year, we already had to stop due to the end of the larvae season. So it seems that we're a little bit lucky for now that the larvae hold due to the relatively long warm weather. It hasn't been freezing yet. But yeah, that's why we try to finish first the Fennery moles, because in case the weather changes and it starts freezing, then we wouldn't be able to finish up the experiment. So that's why we decided first to do the Fennery moles, then the pandemic box, because yeah, otherwise it's better to have at least one experiment finished than two half.
Yeah, I think it's a priority for the paper as well. It seems to be quite cold here. Our winter's carrying on, so maybe you guys are stealing our warm weather. We would like it back.
Well, today it is a little bit colder, so maybe you'll get your nice warm weather back. But it's I think 12 degrees centigrade here now.
I think it's only about 15 here, so maybe we're even.
It's gonna be 17 on Friday, so that's good news.
All right, so I think that's kind of all of the updates that we are gonna have for this month. Anyway, is there anything else that I missed? Sorry.
I'll just chime in and introduce myself. Yeah. Obviously, I'm Christopher Rice. I was the person who done all the pathogenic free-living amoeba screening for you. As you've obviously seen, we have a very strict cutoff when it comes to testing molecules, that we look for items less than 1 micromolar. And the reason for that is we are trying to develop compounds that can actively cross the blood-brain barrier and show efficacy in vivo. In our previous 3 to 4 in vivo studies in mice, anytime we use a compound less or greater than 1 micromolar, we can never achieve concentrations that are effective at inhibiting our parasites and curing the models. So that's one of the reasons why we have such a sort of stringent IC50 to then move forward with lead prioritization of drugs. Obviously, that's more for Naegleria and brain diseases, but there are other diseases that these amoeba cause. But again, we need them at very low nanomolar concentrations to be like active against these amoeba, because generally some of the treatments can last between weeks to months to years. And what we've found is some compounds also can increase toxicity over repeated doses and things like that, you know. So this is how we are sort of looking at our drug discovery strategy from the outside in, and trying to sort of lead prioritize our best compounds forward based off of all these different parameters and specific disease indications that we're looking at that particular moment. So, hi everybody.
Hi Chris, thanks for that.
So there's a couple of action items then from the meeting that I've kind of taken. So one is to incorporate the, I'll call them master codes for the compounds into the Excel spreadsheet using the code system that's already been agreed upon and used in the other open source ones. So is there anyone who would like to put their hand up to do that? Nope.
Are you talking about the coding system in the OSM list?
Yep.
Have we finally agreed on what convention system are we using?
So I think that from
MyOS?
what I gathered from Matt, he wants to use the same one as what's being used in the others, just it would be with the code for OSM instead, but the same numbers and system.
Yeah, I can
There's
do
a
it.
wiki page on for open source data by the S2 compounds on the OSA. It should have everything there. I believe Matt updated that a couple days ago.
If you can find that link, do you mind just popping that in the chat for us?
Yeah, I can do that.
Thanks.
I thought Matt sort of referred to this document on GitHub on the numbering system.
I think there is an issue on GitHub about
Yeah, he
coding
linked
system.
it like a few days ago or a week ago. I don't remember now.
Okay, so we haven't got a system then that we want.
It's all the way at the bottom of this. It was kind of tucked away.
Okay, yeah, I think that's something more for the chemist and the biologist to take on, because I don't know the coding system that well.
So I think the problem will be because we are OSM and I might get confused with open source mycetoma. So I think it's just that 3-letter code that we have at that we would use at the beginning. The open source antibiotics is using OSA. I think if we just make a decision on that, the rest of the system that they're using is fine. It's numbers and letters that.
Maybe we should use OSY as in, Y for, as in myco. The Y,
I thought,
that way we don't mix MY, COSY. That way we don't mix up with malaria, because we do have OSM2, open source malaria.
Again, I just put up a link to what Matt has referred to. The first 4 characters of what he wants to use is MyOS.
Yeah, that's what we were talking about.
With 4 letters instead of
Yeah.
Why did I wish I could ask Matt why OSA, why the antibiotic codes were only 3 when the mycetoma one's going to be 4.
Yeah. Mm.
is MOS already in use? Otherwise, we can use MOS.
Yeah. Not sure. Man, Matt left in the worst time.
Does it have to be 3 characters, or is just something like to make it look good?
I think it's just keeping consistency across all the different open source projects.
Yeah.
To be honest, like 4 letters make more sense.
Yeah, I agree for this one too.
Yeah, I like 4 letters as well. So MyOS is like my favorite.
Okay.
Yeah, so I think, hi, this is Ben. Can you hear me?
Yeah.
Yep.
Yeah.
Okay, cool. Yeah, I think that this, as Wendy said, this is something that someone on the chemist side should take care of. The problem with opening this up as a discussion is that everyone has a slightly different view on it, and we'll just end up talking in circles. Unless I was just someone goes for it, and then everyone else will fall in line behind it, I'm sure.
Okay, it's MYOS, and then the same numbering system that's Matt suggested in that column.
Yeah.
Yeah.
I would
just point out, though, just so everyone's on the same page, right? We're only going to assign these numbers to things that actually exist, right? So concepts, ideas, compounds that are currently being designed or being made should not have these codes. It should only be assigned as soon as something is made.
That is correct.
Okay, just making sure everyone's on board with that, because otherwise it will just become unusable.
Yeah, yeah.
Okay.
So
cool.
for the time until full categorization is achieved, those compounds will just be have an internal code. So it will be HPD for me, for example.
Yeah, exactly.
Until,
the code you're using, or the code that we're using on the OSN and stuff like that, it doesn't matter. The important thing, however, is when it does get given that myos number, or whatever we're going to call it, that there is a second column that has the historic, pre-registration code as well, so that you can link the two things up.
Yeah.
Okay. Good.
So a little naive question being my first talk. What is it you use for compound management? Is it just your Google Doc? And should you look at potentially investing into something for compound managing, like CDD Vault or something like that?
I can maybe reply to that. Matt would definitely reply if he was here. So right now, I think it's Excel sheets, which is not optimal, particularly as the project grows, but it's free and it's amenable for everybody. I think that one of the concerns that we have with CDD Vault is that it's not free, it's not open, and so therefore not aligned with the principles. Matt and I and others are working really closely with some people who are working in the COVID moonshot, who have like a really fantastic solution to all of those problems, which hopefully will be available open source. So we hopefully we'll have something up and running open on this soon.
I'm sorry, Ben, did you ever talk to Open COVID?
Yeah, well, I'm not sure about Open COVID, the COVID moonshot.
Yeah, that's what I meant. Yeah.
Yeah, we're talking with them. Yeah, we're basically going to hijack that. We're going to hijack that code and apply it to everybody. We're trying to take what they've done, which is amazing, and set it up in a way that it can be applied in sort of a one-stop shop for all open projects, whether that's open source malaria or open source antibiotics or the DNDi open source project. Single location, quick crowdsourcing chemistry. That's the current plan.
Well, yeah, it's good for. I think the system was fine when I was submitting compounds, and I saw you on there as well. It was actually pretty intuitive. So that would be nice if we could get something like that.
Yeah, those are the discussions we're having at the moment. We just need funding to be able to do that, but we think we can get some.
Okay, thank you.
That said, Chris, if you have access to CDD Vault is really good at these sorts of things. It just doesn't really tie in right now.
Yeah, currently I have a sandbox version of CDD Vault where we've not fully committed to using it yet because our projects aren't as drug intensive and data merging. But for something like your guys' projects, I understand that it's not open source, but it may alleviate some of the compound management problems that you may start to suffer as you start to grow, so.
It's something that kicks in when it starts growing, indeed, yeah.
Yeah.
But for now, yeah. It's not without its flaws either.
Yeah, of course.
But it's right. Well, no, there are solutions out there that don't read there are solutions out there where the only flaw is the cost, and everything else works. But they cost, unfortunately, a lot.
All right. Is there anything else that we need to discuss? Kimberly, is there an actionable item that any other actionable items that are urgent?
I think the rest of them are like pretty clear on who they're assigned to. It was just that coding one of someone who might want to update our Excel spreadsheet.
Yeah, I can do this.
Thank you.
Thanks, Dimitri. Yeah, so we're coming to the conclusion of this meeting. So is there any other issues that haven't been covered yet that anyone would like to raise?
I think Wendy asked something yesterday on GitHub regarding the compounds.
Did I miss it? Sorry.
Yesterday. Was it yesterday or just a few days ago?
Wendy, do you remember what you asked on GitHub? I think I might have missed it.
No.
I think she's typing the answer.
No, I thought, wasn't it on the coding as well? So
yeah,
that,
it was something to do with coding. You mentioned.
Yeah, because now we generated all these data, but we're also going towards drafting the manuscript. So I think to update all the figures, etc., we need to use the coding we are going to use in the manuscript as well. So that was the part on the coding as well, which I put in GitHub, that it will be good to add an extra column in the Excel sheet so that we have the historical name with the name it's going to be.
So you want to use MyOS coding in the manuscript, is that correct?
Yeah, I think that's the best thing to do, because if the MyOS coding is going to be used for MycetoS, then that should be the coding we also use in the manuscript. Or maybe I'm wrong, but yeah, that seems to me the most logical.
Yeah, I agree. I think that's a good idea.
Hi, this is Ben.
Yes.
Can you hear me? Yeah. It depends. I would say it depends how many molecules are going to be in the manuscript. If the manuscript is in any way has a synthetic chemistry or medicinal chemistry component, then the overwhelming what's the word? Approach is to just use numbers. That's in like 1, 2, 3, 4, 5. That's how it is in every single chemistry paper. So what I would suggest you do is, if you write it, you can use code. One could use codes like, you know, an internal registration code as a placeholder. But when the manuscript is submitted, they should all be changed to just single number ascending numbers.
Yeah, but the issue is that we are also including some of the epichem molecules, so they will have already a clear code, because the epichem library has its own code, of course.
Yeah, what I mean is that for the sake of the manuscript, the convention, the accepted convention in all papers that have any sort of chemistry or synthetic chemistry or medicinal chemistry angle, is that the compounds that are in there are not, you don't use internal codes to signify them, you just use a number.
Yes,
Compound
I know,
one,
but in general, still, it would be good if this code, even if it's 1, 2, 3, 4 in the manuscript, there should be some supplementary file or something like that demonstrating what the end code will be in the MycetoS project itself, so that everybody can always look back, oh, this number in that manuscript is that molecule.
Yeah, so the way that you can do that is that you would put that in the supporting information of the paper. You would have a table, like for example, an Excel table that contains the data. It would also, for example, have linked to how the compound was made, and in there you could put a column that would be the, let's say, the source ID, like an EPA, the epichem number or the MycetoS number or something like that, and that would be attached to the SI of the paper. That's kind of the only way I would. That sort of information would normally be considered.
But they used EPL, for example, in the first Mycetoma paper already. So that's the rationale, like why they want to use MyOS.
That's why I'm saying it depends. It depends what the angle of the paper is. If it's a medchem or synthetic chemistry paper where there are multiple compounds, then I mean, the convention is to use just ascending, just numbers. And I've never seen a paper that doesn't do that. So that's really I think if it is a, you know, if it's a paper that is focused exclusively on the biology and there are only a handful of compounds used, then at that point you can use the codes. But if it's a paper that is talking about the synthesis and a paper that is talking about the structure-activity relationships and those sorts of things,
I'm gonna head off, everybody.
The convention is to use those numbers. Okay, sorry. I couldn't hear who that was.
Well, half the paper. Sorry. Anyway, half of the paper will be about the synthesis of the compounds for things that Hong and Dimitri did. So I think the biological part's only the other half.
Well, maybe we should take this discussion further when Matt's around as well then.
Yeah.
I think that would be a good idea. Yeah, I'm not very experienced in this.
And I would write it using the internal codes. But before this is what I would do. I would then, when you submit it, change it to numbers. And the reason I would do that is if I was reviewing a paper that came in where they hadn't used that convention, I would immediately write back saying you need to use this convention because it's the standard accepted format.
Yeah, because for me, for example, when I put together my my data for the experimental details, definitely in addition to having the chronological order, so number 1, 2, 3, 4, there will also be the name and the code in the experimental detail itself. But yeah, in terms of actually using the code in the main body of the paper or just a sequential number, then yeah, probably need more discussion.
So from our experience in our drug discovery papers, we only use the MycetoS paper for internal. And then when it gets published, you just use a significant number for any of the compounds. So we've done an SAR project where there was probably 100 to 200 compounds involved with that paper. So obviously trying to keep track of all names for reviewers or for readers is going to be difficult. So that's why those numbers are assigned. So it makes it easier for people to review and read it.
Okay, well, I think we should simply look later how the manuscript looks like. And in the supplementary should at least have a table where we can find what number 1, what other name it has, so that everybody can look what is what. Yeah. Because yeah, but yeah, for the chemist, then it's also important if we're going to use the numbering 1, 2, 3, 4, because we have molecules made in London and in Sydney. So you also have to look which molecule will become 1 and which becomes 2, etc.
It goes in order you read it. So the first compound that's introduced is number 1. The second compound that's introduced is number 2. So it just goes like, it doesn't matter where it was made. It's just the order it comes up in the paper.
Yeah, it's the order it appears in the paper. And the way that you keep track of that is that when you write the manuscript, you use a placeholder, for example, a MycetoS number or a combination of different source codes. And then last thing you do once the manuscript is finished is go through and change all of those to ascending significant numbers.
Okay.
Yeah, it's really frustrating to do that, but yeah. I've not found an easy way of doing it yet.
That's how I had to write my thesis as well. So yeah, just a code, and then I just Control or Command H, and then just change it individually later into a number.
Yeah, and so inherently, when you do that, you end up with a table, the sort of table that Wendy's talking about. You have to have that to be able to keep track of it anyway for writing it. So that the sort of table that Wendy's talking about is basically automatically generated by that process, where you can link the identifier number to the molecule number in the paper. Where it gets confusing is if you write multiple manuscripts, because you can have a particular compound that in manuscript A is compound 26, and in manuscript B is compound 3, and that's another thing that would need to be kept track of.
Yeah, so I guess we'll just keep the placeholder for now as we write the manuscript, and then we'll work that out later. We'll just get something written first, I guess. So yeah, so 4 more minutes. Is there any more urgent matters before some of us has to leave and some has to go to bed? Is everyone all good?
Yeah, for me, yeah.
Thanks.
All right. So yeah, so I'm just gonna close the meeting for now. So I hope everyone have a great day / night, and we'll meet again soon as well.
Okay. Have a good sleep.
Thank you. Thank you.
Okay,
Bye,
see you next
everybody.
week.
All right. Bye, everybody.
It was nice to
Thanks,
meet you.
bye.
all.
Bye.
You too, Chris.
Thanks. Bye.
Bye.